Hello everyone and welcome back to PhytoPathoVerse. I'm Malini Ray, molecular plant pathologist with a motto to make plant pathology easy. With ASRB NET 2025 approaching, we are continuing our special series on model papers, each designed with 100 carefully selected questions. So, today we will be moving forward with model paper 8. Question number 1. Anastomosis means is seen in rhizoctonia. It means the fusion of hyphae. Okay. Number 2. X bodies are produced by which type of pathogen? It is virus. Next, virus reproduces by replication, binary fission, budding transformation. It is replication. Next, the characteristic of a pathogen being able to cause disease is termed as pathogenicity. It is the ability of a pathogen to cause disease. Okay. But see, we have a similar term here, that is pathogenesis, which means the process. Okay. The mechanism, that is pathogenesis. But when we talk about the ability or characteristic of a pathogen, that is pathogenicity. Question 5. The external or internal alterations occurring due to disease is referred to as symptom. But then what is sign? Sign and symptom, they are similar terms, right? We normally get confused. So, when we will see physical presence of any pathogen, then we will call it as sign. Next, APS stands for American Phytopathological Society. It is the largest society which has the maximum number of members. Okay. So the word fungicide has derived from which language? Greek, Latin, French or German? The answer is Latin. So it has actually originated from two Latin words. Okay. One is fungus, another is caedo, C-A-E-D-O, caedo. Now the word caedo means to kill. So here it comes, fungicide, killing of fungi. Okay. Question number 8. The disease can be classified based on host plants affected and causal organism involved, right? Plant parts affected, okay. Symptoms produced on the host plants, okay. All the above options are correct. Question 9. What disease of potato caused by Syncytium endobioticum? It is which type of disease? It is endemic disease. We have discussed it in the earlier modules why this is endemic disease, right? Question 10. Which of the following is involved in pathogenesis process? P penetration, post penetration, penetration. All the above are correct. Question 11. The term fungus, Puccinia graminis tritici, enters through stomata. Okay, all the rust fungi enters through stomata. Question 12. A thick strand of somatic hyphae resembling a rod top is generally termed as rhizomorph. 13. A mat of hyphae giving rise to short conidiophores closely packed together is termed as cinema. All these are examples of fruiting bodies. Okay. Question 14. Green ear is a proliferation of flower parts into greenish leaf-like structure generally noticed in. So, see this word green ear. With this itself, we can guess the answer. Here we don't see any option, so I'll give you 3 seconds. You can guess the answer. Okay. So it is ragi and bajra. 15. Question number 15. Measurement of plant pathogen is done by 1. It's dry weight method. 2. It's cell volume method. 3. It's cell number method. All the above answer are correct. All the measurements are used to measure plant pathogen. Question 16. The principal method of controlling plant diseases are avoidance, exclusion, eradication. Again, all are right. Question 17. Most of the virus disease are more virulent when the temperature. So, in this case, it will be 15 to 20 degrees Celsius. Question 18. Potato, dextrose, agar do not contain. So, potato, dextrose, agar is a media. If anybody has started their work on plant pathology, he or she will understand PDA is the generalized media and the first media about what we are taught. So, PDA, potato, dextrose, agar, what it requires? Potato, obviously. 200 gram of potato, correct? Second, what they are telling? Dextrose. So yes, option A, dextrose, 20 gram of dextrose. The third is what? Agar. So yeah, we got to know agar, again 20 gram of agar. So we require dextrose, agar and potato. So which is not required? Beef extract. Question 19. The mechanism involved in spore discharge. It is shooting. Question 20. PR2 proteins. So there are total 19 PR proteins. Last 18 and 19 are not known. So we basically study 17 PR proteins which has their specific roles. But what does PR2 proteins do? It is beta-1,3 glucanases. Question 21. The chain of events involved in disease development, including the stage of the pathogen and effect of disease on the host. This phenomenon is generally termed as disease cycle. Question 22. A compound which prevents fungal growth without killing fungus is termed as. Now for fungicide, we have already seen, for fungicide we have to kill the fungi, right? But here they are saying without killing the fungus. Then it will not be fungicide, it will be fungistatic. Okay, question 23. Asexual spore of the Basidiomycetes fungus is aciospore. Question 24. The fruiting body basiocarp of Basidiomycetes is produced by, it is tertiary mycelium. Question 25. Zero stage of fruiting body in Basidiomycetes. It will be is. Options are Asium, Basidium, Uridium. So can you see any answer of this is correct? Zero stage. Nope. So in Basidiomycetes we have total 5 stages. How we name them? Zero stage, stage 1, stage 2, stage 3, stage 4. Now why these stages started from 0? We have a different story of that. We will cover it in separate video. But the 0 stage is actually pycnospore, which is not here in any of the options. So it will be none of the above. Question 26. Sterile threads arise at the base of the perithecium, upward direction between asci. At the base, we get to see paraphysis. Question 27. Cellulosic cell wall pathogen. Cellulosic cell wall means what? They belong to Oomycetes. So Oomycetes fungi, Pythium, right, they belongs to Oomycetes. Phytophthora, again, yes. Bremia, yes. So all of the above. Question 28. the fungal pathogen containing chitin in their cell wall. Now if they have chitin, then it will belong to higher fungi. So among the options, Phytophthora, nope, we have just seen it belongs to Oomycota. Sclerospora, again downy mildew genera, belongs to Oomycota. Next, Peronospora, again Oomycota. So Clavices purpurea, it belongs to Ascomycota, which is a what? Higher fungi. And higher fungi, they are having chitin and glucan in their cell wall. Next, question number 29. The sexual spore of Pythium aphanidermatum. It will be oospore. Same reason applied. Pythium belongs to Oomycota. Question 30. The sexual spore of Rhizopus. It will be zygospore because Rhizopus belongs to Zygomycota. Next, 31. The sexual spore of Gibberella fusicori is ascospore because Gibberella belongs to Ascomycota. Simple. Question 32. Vegetative spore of Fusarium. Vegetative spore of Fusarium. Now Fusarium produce how many spores? They produce 3 different types of spores. One, Microconidia, second, Macroconidia, and the third, Chlamydospore, right? Even if you don't know it is a vegetative spore, but only one option we have it here, that is Chlamydospore of a type of Fusarium producing spore. Okay. Question 33. Long distance moving spore in rust. That is Euridiospore. Euridiospore can travel very long distance, even country to continents, and that long. Question 34. Clamp connection is most common in case of smut. So yes, anybody can be confused between why not rust, why smut. Because rust also belongs to Basidiomycota, right? And clamp connection is a characteristic feature of Basidiomycota. Okay, but if you see the difference between rust and smut, then you will understand, yes, both belong to Basidiomycota, but clamp connection is absent in rust fungi, but they are present in smut. Try to remember that. Question 35. Clamp connection is most common in case of Eustylago tritici, which is a smut fungi. Got it? 36. The spore responsible for wheat rust in India, that is Euridiospore. Why they have framed the question like in India? Because for wheat rust like this, the stem rust or black rust of wheat, they require alternate host. They are heterogeneous in nature, and the alternate host is Burberry, which is not grown in India. Now to complete its cycle, they require 5 different types of spore, right? PAUTB. But in if there is absence of Burberry, then how the disease cycle will complete? Correct? But again we get to see black rust. So in so many places, mainly in the southern India, we get to see black rust. Then how it happens? That's when Euridiospore comes in. That's why Euridiospore is also called repeating spore. So it can be used as a new inoculum for starting a new disease cycle. Understood? Okay. Question 37. Dikaryotic spores smooth bond in chains in rust fungi is teleospore. One reason is because teleospore is dikaryotic spore, correct? The second is chain, the formation of chain, because teleospore is bicelled, so they are connected together and it looks like a chain. That's why. While Euridiospore and this pycnospore, they are single celled. Okay. Question 38. The multi-celled spore In case of rust, again it will be teleospore. Okay. Question 39. The dormant spore, which is vegetative spore but not asexual. Okay. It will be chlamydospore. Okay. Because see, all the other options like sclerotia or mycelium, they are not spores at all. They are hyphae. Right? So the answer is chlamydospore. And it is also vegetative or fusarium. Next, 40. Gametangial copulation mainly observed in. Now, there are different type of fertilization that takes place. Fertilization in sense of reproduction that takes place, right? So, there are gametangial copulation, gametangial contact, and spermatization. Like these different types are there. So, what we get to see is that according to the options, Phytophthora belongs to Oomycota, Pythium, Oomycota, Claviceps, Ascomycota, right? In both these classes, Oomycota and Ascomycota, the type of reproduction that we get to see is gametangial contact, not copulation. Okay, gametangial copulation we get to see in case of Zygomycota. So, all the options are wrong. So we will add one more option here, that is none. Okay, so none will be the option number D, and we will strike this. Okay, so option D is none, and none is the correct answer. Okay, question 41. The herbarium Cryptogamy Indian Orientalis was first created by E.J. Butler, and you know where is it present right now? It is in New Delhi. Question 42. The sterilization temperature in autoclave for 15 minutes, 15 to 20 minutes, 15 psi for 20 minutes, that's what we study. 121.6 degrees Celsius. This will be the precise temperature. But when we will not have this option like 121.6 degrees Celsius, we will go straight to 121 degree Celsius at 15 psi for 15 to 20 minutes. Okay. Question 43. Complete root phanerogametic parasite. The answer is Orbanshi. Just remember it, learn it, mug it up. All these options are examples of phanerogametic parasite. Okay, we just need to know which one is complete root parasite, partial root parasite, complete stem parasite, and partial stem parasite, right? Like next we have complete stem parasite. It will be daughter or cascata. Next, again the similar question, complete root parasite, it is Orbanshi, already discussed. Next, they asked partial stem parasite, it will be mistletoe or Lorinthus. Okay, question 47. The genus directly germinates in the family Perennoporaceae. In the family Perennoporaceae, we get to see the genera of downy mildew. Okay, let's go for the options. Bremia, Perennospora, Perennosclerospora. All are genera of downy mildew. So the answer will be all of the above. Next, question 48. The slime molds comes under the class Myxomycetes. Question 49. The plasmodium or plasmodium-like structure is seen in division, again Myxomycota. Question 50. The genus Olpidium and Physoderma belongs to the order Chytridells. 51. Which one of the division belongs to lower fungi? It is Myxomycota. Question 52. Bread molds belongs to class Zygomycetes. Question 53. The mycorrhizal fungi producing spores singly in soil or on sporocarp containing zygospores, chlamydospores or sporangia. It belongs to class, okay, it will be endogonals. This is a genus of mycorrhizal fungi that we use. We call it endogen, which came recently in our ICAR AICE-JRF SRF PhD exam. So this is one of the statement that came in the exam. One commonly that we use is glomus, and the second option was endogen. So remember it. And the class, this is a kind of order that they have given the options, endogonals. Question 54. The imperfect fungi wherein sexual reproduction and structures lacking or unknown comes under division. Imperfect fungi equals to Deuteromycotina. Question 55. The rust and smut fungi belongs to the class Basidiomycetes. So answer will be option A, Hemibasidiomycetes. Question 56. The club-shaped fungi is the special characteristic of subdivision. Again, Basidiomycotina. Question 57. The wood decaying and root rot fungi grouped under class, which one will it be? It is Hymenomycetes. Next, 58. Nucleic acid content of plant virus. It consists of 5 to 40%. This percentage is important. Similarly, we will see the protein content of plant virus consists of 60 to 95%. Nucleic acid is 5 to 40 and protein content is 60 to 95%. Next, 60. This spherical shaped virus contains spherical, when we make a circle, the space is more, right? So they contain higher percentage of nucleic acid. But wherein we have elongated shape plant virus, means like this, elongated shape. So the space between here will be less, right? So there we will have lower percentage of nucleic acid. Next question, 62. Nucleic acid of most of the plant virus consist of single-stranded RNA. 63. RNA consists of ribose, phosphoric acid and base. Correct? All the above are correct. 64. DNA consists of deoxyribose, phosphoric acid and base. Again, all are correct. 65. Purines are made up of adenine and guanine. So, both A and B are correct. One more question should have been there. As RNA is there, DNA is there, purines are there. So, again, one more question, it can be pyrimidines. So, pyrimidines are made up of what? Nucleotides that we talk about, they have, we normally have 4 nucleotides, right? A, T, G, C in DNA. Adenine, thymine, guanine and cytosine, correct? So purines are made up of adenine and guanine, means A and G. So pyrimidines will be made up of thymine and cytosine. Okay, so one more question can be made of this. Coming to question number 66. In RNA viruses, thymine is replaced by uracil. Okay, so in case of RNA viruses, what these pyrimidines will be made up of? just now we have seen, right, thymine will be replaced by uracil. So in case of RNA viruses, pyrimidines will be made up of uracil and cytosine. Alright. Now question 267. The order Homoptera includes, this is an insect order which consists of aphids, leafhoppers, whiteflies. All the correct. 68. The insects which feed on infected plants for several minutes to hours to few days before they accumulate the enough virus transmission. This type of viruses are called semi-persistent. Why? Because they have asked like for minutes to hours, several minutes to hours. If it was less than that, like for few seconds to minutes, then it would have been non-persistent. And if it was asked like for days and months, it will be persistent. Okay. So these are the mode of transmission that we study in virology. And also we get to see few questions from entomology like insects, because insects are the one of the mode of transmission of plant viruses. That is the reason. Now question 69. The stylet borne viruses persist in the vector for only few minutes to hours are known as non-persistent. One of such reason that you can tell it is non-persistent because they are stylet borne. If instead of stylet borne, they would have asked fogged borne, so it would have been semi-persistent. Okay. Okay. Question 70. Virus which persist in vectors for few days are known as persistence. Now it comes to persistent. You know, I understand if anybody is seeing this and they are new to agriculture or plant pathology, so no need to worry. It will get easier when you will understand the mode of transmission of viruses. Okay. Just go with the flow. Question 71. Viruses which multiply in respective vector known as propagative, multiplying virus. Answer is both A and B. Question 72. Leafhopper and plant hoppers transmitted virus causes disturbances in plants that arise primarily in the region phloem. Okay. 73. Fungi which transmit plant viruses are, yes, fungi is also a mode of transmission of plant virus. Olpidium, Brassicae, Polymixa, Graminease, Spongospora, Septerinea. All these are fungi which transmit plant viruses. All are correct. Now 74. Nepoviruses are. Now why this term nepo? In every question and every word there are so many things that we can study and we get to know something new. Nepoviruses are the nematodes. Okay, the nematodes which transmit plant viruses. They are classified into two categories. One is nepovirus and another is netovirus. Okay, so the type of viruses which is transmitted by nematodes that belongs to nepovirus, they are grave fan leaf virus and tobacco ring spot virus. So both A and B. The first transmission that we get to see with nematode, that is grape fan leaf virus transmitted by Xiphenema species. Okay. 75. Netoviruses are tobacco rattle virus and P. early browning virus. Both are correct. They are normally transmitted by the nematode species such as Trichodorus, Paratrichodorus. Okay, and this for nepoviruses, the nematode species which transmits nepoviruses are Zyphonema index, for grape and leaf fan virus, also Longitudinus, Paralongitudinus. All right. Next, question 76. Plant virus group containing negative single-stranded RNA. Options are rhabdovirus, tospovirus and tenuivirus. All are correct. Question 77. The plant virus containing single-stranded DNA virus. That is banana bunch-it-up virus, correct. Tomato leaf curl virus, again correct. Third, pumpkin yellow vein mosaic virus. All the above options are from begamovirus, Gemini viridae family, right? So, Gemini virus, they are having single-stranded DNA. 78. The genomic content of virus. It is double-stranded RNA. Question 79. The genomic content of tospovirus is positive sense single-stranded RNA. Question 80. Use of mild strain for protection of plants against to severe strain of same virus. The process is called cross protection, and it was given by McKinney, in which virus it was first discovered in TMV, but nowadays it is most commonly used in case of citrus tristeza virus. Okay. Next, 81. The beetles transmitted viruses. Beetle transmitted virus. Okay, thymovirus, comovirus, bromovirus, all are correct. Question 82. The mite species which transmit plant viruses, Asaria kajani, Asaria tulipi, both are correct. Question 83. The process in which conversion of RNA to DNA, it's called. Normally in central dogma, we get to see DNA to RNA, that is called transcription, and RNA to protein, the process is called translation, right? But when we see ulta, like opposite, from RNA to DNA, then we will call it as reverse transcription. Okay. Question 84. Circular nature of mini viroid was shown using. Normally, even viroid we are not able to see with normal microscope. Even for viroid to observe, we are in need of electron microscope. And in the question they have asked mini virus, so obviously it will be electron microscope. Question 85. Genome content of viroid. It is single-stranded RNA. 86. Antibody is made up of only proteins. Antibodies are made to fight with antigen, right? But what this antigen will be made up of? Now, antigen can be any foreign particle. It can be anything. So, in this case, it can be protein, it can be polysaccharide, it can be lipids. So, all of the above. Question 88. Antibodies are secreted by B lymphocyte. Sorry, it's B lymphocyte. 89. Antibodies are divided into, they have like 5 classes. I don't remember right now. It was something IgG, IgA, IgE. There are like this 5 classes are there of antibodies. Question 90. The measure of overall stability of complex between antibodies and antigen. When they will ask like overall stability, it will be avidity. When they will ask measure of strength, it will be affinity. When they will ask concentration, measure of the concentration of specific antibody, it is titer. Okay. Question 93. Matthew's Plant Virology is written by Roger Hull, which is an important book for plant virology. 94. The density gradient centrifugation as a method for purifying virus was first developed by Braque in 1951. Question 95. A virus having multipartite, like they have multiple partition, it will be alpha-alpha mosaic virus and they are tripartite. Okay. Question 96. The genome size of potivirus, it is 8.5 to 10 kb. 97. A citrus tristeza virus belongs to Clostridium, and again this Clostridium, they are the longest plant virus that we know of. 98. Bigomovirus are tomato leaf curl virus, cotton leaf curl virus, bhindi yellow vein mosaic virus. All are correct. Question 99. Papaya ring spot virus transmitted by several aphid species in the non-persistent manner. Question 100. Last for this module. Zucchini yellow mosaic virus is transmitted by aphid. Okay, that is the end for today. Here we got to see many questions from plant virology. Okay, so yeah, there are more questions from that can come from plant virology, but this module is mostly from plant virology. Okay, let's see what next module brings. That's all for today. We will be back soon with a new model paper. Till then, keep learning, keep growing. See ya.